Good morning, children. Welcome back to the lecture series for Zoology Intermediate first year. I am Vishnu Priya, Principal, TSMS Palamokla. Till date, we have completed the phyla Porifera, Cnidaria, Ctenophora, Platyhelminthes, Nematoda, Annelida of the unit Animal Diversity 1. And in our journey of exploring the various phyla of the animal kingdom, we arrive today at the largest phylum of the animal kingdom, Arthropoda. Perhaps there is no house without insects that belong to this phylum. The unending list of encompasses the spiders, cockroaches, prawns, crabs, scorpions, butterflies, etc. This phylum is the first to have wide varieties of species with divergent colors and distinct shapes. It comprises 80% of all known animals. Arthropods have a long history. They have been on Earth since the early Cambrian period, around 500 million years ago. One of the extinct organisms, trilobites, are the oldest known arthropods. Their fossils have been very useful in understanding life on Earth during the Cambrian period. The earliest animals are all aquatic. Arthropods were the first animals to adapt to life on land from living in the water. The common ancestral arthropod is assumed to have an identical pair of appendages on each segment, then went through lots of evolution to become what we see the arthropods these days. Today, we have with us a new speaker, Miss Suneela C. KADAM, PGT Zoology, TSMS Nedunur Kandukur. She has received the Mandal Best Teacher Award. She has a vast, accomplished teaching experience of 20 years and has been with us since the inception of model schools. Now, Suneela Madam will take you to the study about arthropods, their classification into subphyla and classes, and their general and unique characters. Suneela Madam, please go ahead with the lecture. Welcome, Madam.
Good morning, Madam.
Good morning, Amma.
So, at the onset, I want to thank Madam Vishnupriya Madam, Madam who has initiated this initiative, our Manjari Madam who has given lot of support and suggestions, our AD Sir, Satyanarayana Reddy Sir, who has taken this initiative to bring the education to the students, the rural background students who do not have much data and all that, so that they can understand, they can come in contact with the studies with this YouTube videos. This initiative is highly applaudable. Thank you very much, Madam. With this, I'll share the lecture now. Yeah, I'll share the I'll start the slideshow now. Good morning, children. We all, just now as Madam has said, we are going to read about a very important phylum. The phylum name is Arthropoda. Arthropoda is the largest phylum. Now let us look at the examples of Arthropoda. In Arthropoda, many of the animals which we consume for food, these are the animals which we consume: lobsters, shrimps, and shrimp. These are the food which we eat. And animals which are like pests, the centipede, millipede, and grasshoppers, these are also included in this phyla. Another examples are, these are the insects which cause diseases. Of course, dragonfly also is a pest, but then ants, scorpions, bee, mosquito, these are the insects which you see around you. We are going to read about the largest phylum, which includes so many animals which are around you. Now, with this, let us come to look at the phylum Arthropoda. These are the subphylums which I am talking about: Crustacea, Myropoda, Chiliformes. But the largest one is Hexapoda, that is, which includes the largest class, that is Insecta. This uh, largest phylum name has come because of the group Insecta. We should be very much. Now, let us come to the introduction here. Before that, let us come to the objectives of this session. What we are going to learn is in this session. First one is we are going to read about the general features of the phylum Arthropoda. We are going to read about the general features. What are the characteristics of the Arthropoda? After that, each class, when we talk about NEET and MSET exam, classes are more important. We are not going to read about the phylum. What will be more important is the classes here. After that, economic importance of the phylum Arthropoda. This is also very much important. Why economic importance is important is because the silk which you get, we are getting from Arthropoda, silkworm. The honey which we consume, which helps in the farmers for the subsidiary income, that also comes from the phylum Arthropoda, Apis mellifera, that is the honeybee. Let us come to the next one. Let us look at the introduction of Arthropoda. Arthropoda is an invertebrate phylum. When we say invertebrate phylum, this phylum, invertebrate phylum, here we have the animals which do not have vertebral column. No vertebral column. I do not want to go fast. I'm going very slowly, children. So, these animals which are invertebrates without vertebral column are included in the phylum Arthropoda. In these invertebrate animals, they have an exoskeleton. This exoskeleton is present outside the body, and this is made up of chitin. I'll come back to this later in the session, but you have to remember that this exoskeleton is made up of chitin. They have a segmented body. This body which is internally segmented. So there are repeating units in our body, in the body of arthropods. Next one, they have paired appendages. These are the extensions of the body which are paired. There are two in number on both the sides, right and left side. They have this appendages. After that, let us come to the next one. You have jointed feeding appendages. The same appendages are used for feeding. Now, when you look at the cockroach here, you can find this. These are the sensory antennae, but here you can find pedipalps which will help in feeding. So, these appendages will help in feeding also. They are very much active and energetic animals. This is the most active invertebrate group. They can walk, fly, burrow, jump, and considering the size, they are very fast. They can fly. We can look, when I have given the example of cockroach here, this cockroach can fly. In the same way, you can see the house flies, mosquitoes, which are included in this phyla. Even they can fly. So that is happening at the rate of 30 miles per hour. And the next, some insects also run at 10 miles per hour. So they can run also. After this, let us come to the next one. Why we have to read about Arthropoda? What is so special about Arthropoda? Arthropoda is the largest phylum. It is the largest phylum in the animal kingdom. We think that human beings and the other animals which we see around us, they are more in number, but what the number is more, but the variety which is there is more in this phylum only. That phylum is Arthropoda. After that, let us come to the largest class. The largest class is Insecta. Next one, two-thirds of the animal kingdom is made up of Arthropoda. Two-thirds of the animal kingdom comprises Arthropoda. 80%, that includes the 80% of the species. When I say 80% of the species, that is equal to 11 lakh 70,000 species. That is a huge and vast number. Next one, let us come to, this is a very important point. What you have to remember is that the scientist name Carl von Siebold. This is the scientist. The name of the scientist is Carl von Siebold. This is the person who gave the name Arthropoda. These are the points you should remember for competitive exams. Either it is NEET or MSET. These are very much important. Arthropoda name was given by Carl von Siebold. I am repeating it twice or thrice. Important points I am going again and again. So these are the unique points of Arthropoda. Another unique point of Arthropoda, look at the various examples of Arthropoda once again. This is Palemon. This is the common prawn which you can see. Then you have, this is the crab. Scientific name also you have to remember. Each and every organism, scientific names are important. Given we are biology students of zoology should remember this. This is Palaminus. This is scorpion. Next one is, this is centipede. Now this is cyclops. This is water flea which we see on water. This is the water flea. Now let us come to the general characters of Arthropoda. General characters of Arthropoda. They are bilaterally symmetrical. Bilaterally symmetrical. When you draw, I'll complete this slide and I'll explain to you. Look here. When you look at, this is the principal axis. This axis which is there, this is the principal axis. Now this is the principal axis. At this principal axis, when both the sides are form a mirror image, when a line passes through the principal axis and both the sides, they form the mirror image, such kind of symmetry is called bilateral symmetry. So this animals are bilaterally symmetrical. Next one, they have triploblastic. They are triploblastic. That is, this triploblastic is the germ layer. This is the germ layer. Triploblastic means the germ layers, and these germ layers are, there are three. All three are present in the Arthropoda. They are the endoderm. I am not going to write the complete this because the space is very less. Endoderm, ectoderm, and sorry. This is not ectoderm. Middle layer is mesoderm. Middle layer is mesoderm. Mesoderm. And then you have the last one, that is ectoderm. These three layers are present in Arthropoda. So remember, in Arthropoda, they are triploblastic and they have three germ layers when we say this. Next one, they are metamerically segmented. This point I am going to highlight again in the next slide. So I am not going to say much about this. Next one is the schizocelomates. Let me remind this once again. Schizocoelomates are the animals which have true coelom. There are only 3 phyla which are schizocoelomates. You have Annelida, which you have read earlier. Annelida, I am writing here. Schizocoelomate animals are Annelida, Arthropoda, Arthropoda, and the mollusca, which follows Arthropoda, Annelida, Arthropoda, and mollusca. These are the 3 group phylums which are schizocoelomates. Schizocoelomates. After that, let us come to the metameric segmentation. I was going to tell about this. Metameric segmentation is the segmentation which is true segmentation. Metameric segmentation is true segmentation. Here, there is an external segmentation. When you look at the body, outside where how many divisions are there? Same divisions we can see inside the body, that which includes the internal segmentation. The body is divided into many number of segments. In this, you have, so this is shown by Annelida, Arthropoda, and Chordata. Now look here. Metameric segmentation, examples I have given. Arthropoda, Annelida, I have given earthworm. Next one, in Arthropoda, we have taken a lobster here. And in Chordata, we are taking the example of fish. There is a repeating unit. The same thing you can see in vertebral column also. There is a repeating unit here. This is what you have to observe. This is present in Arthropoda. Next one, let us come to the next one. Arthropoda, one more important thing we have to remember about arthropoda is that for the first time, striated muscle came in arthropoda. For the first time, striated muscles were seen in arthropoda. This striated muscle is also called voluntary muscle. Arthropoda is all, the striated muscle is also called voluntary muscle. Now, voluntary muscle, what is the use of voluntary muscle? It helps in the movement of the organism. The control of the movement is under the will of the organism. That is the reason that striated muscle is very much important. After that, let us come to the coelom. I was talking to you about the coelom. Here you have schizocoelom. This schizocoelom, before I explain schizocoelom in detail, let us look at the next one. Here, this is the striated muscle. Next one, schizocoelom. Yeah, this schizocoelom, this is the 8-cell stage after the division of the zygote. Before here, I'll write here, this is the zygote which divides into 8-cell stage. This 8-cell stage, it goes into the next stage, which is called the blastula. This blastula, it consists of a cavity which is called blastocele. The cavity which is present in the blastula is called blastocele. Now, this blastocele, you can look here. Here you can see that there is a blastocele. This blastocele is called primary cavity. This blastocele is called primary cavity. And this primary cavity is continued in the arthropods. In the Although it has a schizosylum which is lined by the mesoderm, this blue color lining is called mesoderm. Okay, mesoderm is lining the coelomic body cavity on the, this is the gut and the body wall. The the body wall, this cavity, the uh, body of an organism in the body of the arthropods. Here it has a true coelom. This true coelom is made up of the true coelom is made up of mesoderm, which is lining the cavity. When it is not present, when it is reduced, then it is reduced to the excretory organs and the genitalia. It is reduced to the excretory organ and the genitalia. So the blastocele, which is a primary cavity, is continuing in the arthropods. Next one, hemocoel. Hemo, anything related with blood is included in hemocoel. And this is the coelom, which is filled in the cavity which is called the sinus. The cavity is called a sinus. Cavity is This cavity is called the sinus. Okay. Now let us come to the next one. Yeah, I was talking about the hemolymph. Hemolymph is Hemolymph is the fluid, the blood which is present in the lymph cavity. Next one, you have the blood which is colorless, or either it is made up of a copper containing pigment which is copper containing pigment which is hemocyanin, which is the respiratory pigment. Now let us come to hemocyanin. Hemocyanin, as I said, it is a respiratory protein. It has copper. It binds to oxygen. It binds to the oxygen. It transports oxygen. It is present in arthropods. Now, let us come to the hemocyanin. Hemocyanin, it is bigger than oxygen. It is bigger than hemoglobin. Now, hemoglobin, it carries only, look here, it carries 96 molecules of oxygen. When you compare this with with hemoglobin, when you compare this with hemoglobin can transport only 4 molecules. So this is a better, it is a more efficient respiratory pigment. Where is this present? It is present in crustaceans, arachnids, and horseshoe crab, that is limulus. Next thing, when it is deoxygenated, it is colorless. That is why most of the time the blood in arthropods, we say it as colorless. But on oxygenation, it becomes blue in color. Let us look at the animals which have hemocyanin. The horseshoe crab, arachnida, that is spiders, and then crustacean, that is lobsters. Let us come to the difference between coelom and hemocoel. Just like you can see here, the cavity is the main body cavity. In the hemocoel, it is the primary body cavity because it is formed from blastocele. The organisms which are forming the organisms which are forming are annelids. They have they are the primary body cavity is present in annelids, echinoderms, and choroids. whereas the blast this kind of the body cavity is reduced and it is filled with fluid that is present in hemocoel. They are found in mollusks and arthropods. Now let us look at the fluid that is a coelomic fluid in the true coelom. Then you have the hemocoeloic fluids. We are concerned more about arthropods, so I am going to talk about arthropods only here. You have the primary body cavity. Next one is the, when you look at the lining here in the coelomates, you have the mesoderm which is lining here, which is called the coelomic epithelium, but here it is made up of the basal lamina or the epithelial sheet. Next one, hemostasis. We are going to talk about hemostasis. When I talk about hemostasis, uh, any organism, all the living organism, whether it is an amoeba or it comes till, whether it is an amoeba or the huge animals like blue whale also, all the organisms, they try to maintain constant internal environment. What it is made up of fluid, so that environment it has to maintain, which is called self regulating process. The process in which it is trying to maintain the internal environment is called self regulating process of the biological systems. Biological systems are nothing but the living organisms which tend to maintain the stability. The word here is, the key word here is stability. Stability is very, very much important here. Any organisms try to do stabilize itself, so that the cells which are there in the organism, all the organisms are made up of cells, and these cells, they can function at the optimal level. That is what is actually the reason why, to maintain hemostasis, organisms, they go for the various kind of roles. So next one, the exoskeleton. Exoskeleton, what are the other functions which are done because of this hemostasis? Let us look here. There are three things which are done here, and they are the uh, they are the circulation. These are the various processes which will help in keeping the constant body environment. Here we have circulation. Next one, you have the respiration. All the are basic function, basic life processes which will help in getting substances inside or excreting substances out. So these are the three things which will help in the the maintaining of the constant body environment. We will come back to that, but before that, let us come to the skeleton. The skeleton of the we were talking about the skeleton of the arthropods. They are made up of exoskeleton. This exoskeleton is made up of chitin. Chitin is a polysaccharide which is made up of N-acetyl glucosamine. Acetyl glucosamine. Chitin is made up of N-acetyl glucosamine, and then it is sclerotized. What do you mean by sclerotized? When there are waxes added to that, some proteins are added to that, and some chemicals are calcium carbonate, such kind of chemicals are added, then it is sclerotized. As itself, chitin is very soft. To make it hard, it is sclerotized. So the exoskeleton is hard because of the sclerotization. In the arthropods, there is an external covering, external exoskeleton, which is sclerotized, and it is made up of chitin. The main material is chitin. So what is the use of the chitin? Chitin helps in protection, as well as it is it acts as a waterproof for life. What do you mean by waterproof of life? It helps in water conservation. We all know they are terrestrial animals, and it helps in water conservation. In Annelida, conservation. In Annelida, we saw that the earthworm has a moist skin. So you imagine the amount of water it has to, it is going to send into the atmosphere. But here that kind of problem doesn't arise because the exoskeleton is present. Now, in the relation with the exoskeleton only, we are going to look at the jointed legs, which are the appendages. These are the joints which are present here, which gives the name arthropoda. Artho, jointed legs. Arthritis, disease which affects the joints, is called arthritis. Now, let us look at the legs. These are the two appendages which are present. The appendages can be given the name of biramus or uniramus. When you say bi, it is two. Bi means two, and bi means two. Look here. This is biramus. Biramus means two. The appendage is divided into two parts. This is called biramus. Whereas when you look at this uniramus, it is made up of only one part. That is why this is called uniramus. You have biramus and uniramus. Let us come to, yeah. Now let us come to the next one, tagmatization. When we say the division of the body, there is a tagmatization is present. The arthropods, just like the annelids, they have segments. When we are reading about annelids, in annelids, we have segments which are present. There is a repeating unit, and that repeating unit is called annulus. That is the reason why the name annelida has come. But here in arthropod, the body is divided into basic structure. In the same way, like our body, we divide our body into head, trunk, and limbs. In the same way, in arthropods also, there are tagma present. Here in annelids, there are separate rings are present, whereas in annelids, we have the segments, they fuse together and they form a group which is called tagma. For example, head is one tagma, thorax is one tagma, and abdomen is one tagma. So the body is basically divided into three parts, and that process is called tagmatization. Tagmatation. Okay. So now you look here. There are two kinds of division which we can do in arthropods. Either, this is an example I am giving of prawn, which is a crustacean. In crustacean, maximum animals we can see is they have a cephalothorax. Cephalo. Cephalo is the, sorry, I have, yeah, I have gone here. In cephalothorax, let me highlight here. Let us look here. Cephalothorax. Cephalo, head. Thorax. The chest region is called thorax. Thoracic vertebrae or thoracic this. In the same way, here you have the chest and the head region are fused together to form the cephalothorax. And the next part is the abdomen. Let us come to the next one, that is the insect. This is an example of insect. We can clearly see that there is a demarcation, there is a head present, thorax is present, and abdomen is present. Head, thorax, and abdomen. Now, let us come to the next one, the nervous system. When you look at the nervous system, here, when we talk about nervous system, you should always remember that nervous system of invertebrates and vertebrates. Now we are going to look at the difference between the nervous system of vertebrates and invertebrates. All these points are very much important in view of competitive exams. So, one thing, look here, I have clearly demarcated the various systems here. This yellow color system is the cardiac or the circulatory system. Green color is the digestive system. Yellow color is the cardiac system. Next one, this is the digestive system, and the blue color is the nervous system. So let us take the example of human beings. When you look at the example of human beings, our heart is ventral, but here in the arthropods, which are invertebrate, special significance is here. The circulatory system is always dorsal, whereas the nervous system is ventral. You have a ventral nervous system. This is what is seen in the look here. I will give you a magnified view here. The nervous system is ventral. You imagine a car. When you look at the car, the lower portion or the wheels, they are the ventral portion. The roof portion is the dorsal portion. This way you can remember the heart is dorsal in position, whereas the nervous system is ventral in position. Whereas it is opposite in us human beings, where you have the heart as ventral and the uh, nerves is dorsal in position in the vertebral column. Let us come to the next one. The nervous system is basically annelida type. What do you mean by annelida type? You have to remember both. Look here, you have a brain. Next, you have a brain is present in the form of a ring. When there is a brain which is present in the head region, but it is in the form of a ring, then that is called the annelida type of nervous system. But when you look at our brain, we have a solid brain. Here it is in the form of a ring. Next one, there is a nerve cord, and this nerve cord is a double. Again, let me magnify here, and you can come to see here. You can see that there is a ring-like structure which is formed because of the connective here. Circumesophageal connective. Esophagus, it is just near the esophagus. And then you have a nerve cord, a ventral nerve cord. Ventral, look at the word here, ventral. And there is a nerve cord which is just like a ladder. Nichana laga, it is like a ladder. Here you can find that there are a group of cell dot or a group is there. From there you can find an extension that is called the ganglia. I'll come back to the ganglia again here. This ganglia is made up of group of the nerve cells. When all the nerve cells are concentrated at one point, then that is called a ganglia. From the ganglia you have the extensions which are the axons. When they are formed as a group, then that is called a nerve. The word which nerve which we say is coming because of a group of axons. After that, let us come to the next slide. There is a well developed cephalization, cephalic, anything related. Zoology is the beauty of zoology lies in the words. If you understand the words, you can remember them very well. Cephalization, head region. There is a very well developed head present in arthropods. This so due to civilization, as you must have read in earlier chapters, due to civilization, what is happening is all the nerves, the sense organs, and all these things are concentrated on the head, and they can perceive the danger. very quickly, or they can react to the danger very quickly. So, more advanced organisms show a well developed head. Civilization is a sign of advanced evolution. Next one, let us come to the next one. It has number of sense organs. What are the sense organs? In arthropods, we saw eye. Here also, we are going to look at the different kinds of sense organs. In that, you have the brain. Next one, you have the sensilla. The brain. Now we have the eyes. There are two types of eye present. You have a simple eye and a compound eye. Simple eye is made up of, simple eye is used only for perception of light. Simple eye only helps in perception of the light. That is the reason why when you look at cockroach, as soon as you put a light, it will vanish. That is because of the ocelli, which are usually present near the antenna. Next one, sensory organs. There are four, we am going to read about the four sensory organs here. You have the antennae, the compound eye, and the statocyst. We have read about the eyes, compound eye. Compound eye, many number of eyes are present. When we read about the type study of arthropoda, we are going to type study of cockroach, Periplaneta americana. We can look at that again. After that, let us come to the statocyst. Statocyst, I'll magnify that and show you. Statocyst is the region why where this is the balancing organ. Statocyst is the balancing organ which will help in keeping the organism in a state, in a equal position. Next one, let us come to the next one. In arthropods, we see ecdysis. What do you mean by ecdysis? Ecdysis is the shedding up of the external chitin, which is formed by the skeleton. Next one, the digestive system. We are going to read about the system in this, the digestive system. When you look at the digestive system, again let me enlarge here. Here, digestive system is a complete digestive system. In arthropods, the digestive system is a complete digestive system. How can you say it is a complete digestive system? Because it has a mouth as well as an anus. If you look here, a complete digestive system consists of a mouth and an anus. And then the digestive system is divided into foregut, midgut, and hindgut. You cannot say that this is a this is present in all organisms. This is present in all organisms. In arthropods, maximum arthropods, we can find the digestive system, which consists of jaws, pharynx, and gizzard. Next one, you have the midgut also, which has CK and digestive glands. One more thing you have to remember is that there is an hepatopancreas. Hepatopancreas. There is an organ which is doing work of both liver as well as pancreas. So the evolution has started already in Annelida. Here there is a little bit more development, and here this is shown here. Next one, let us come to the next part where the arthropods, same in the digestive system only, you have the spinning apparatus which is shown in Arachnida. This is very much very much prominent in the spiders, where they produce silk with the help of spinnerets. Next one, let us come to the next hemostatic measure and the process, which is the respiratory system. The respiratory system, in arthropoda, we see gills, which are the respiratory organs. Now, when gills are present, such kind of respiration is called branchial respiration. So, gills are because they are the typical of aquatic animals. They are the evaginations. They are the foldings which are present here. Now, blood, which is 21% in very little amount of oxygen can be taken up by the gills. And then They are delivered directly to the tissues from the oxygen. Okay. Next, they are in crustaceans. Crustaceans, you have the gills. Next, in arachnids, we have the book gills. Look here, look at the arthropods. We are looking at uh, the respiratory organs. These are the trachea which are present in insects. Trachea is a tubular structure. You can see here there are tubes present. These are the tubes. Next one, this is the book lung. Just like the pages in a book, they have the book lungs. Now, what do you mean by book lungs? These are the pages. This pages where the air will go in here. This is seen in arachnida. Next one, these are the gills which are present in crustacea. Book lungs of arachnida. These are the gills which are present in Palaemon, that is the prawn. Respiratory organs in Arthropoda, you have crayfish and prawns, you have gills. Insects, you have trachea. This is what I have said earlier. Next one, mites. Mites are the organisms which again belong to arachnida. They do not have a proper respiratory organ. So what happens is they respire with the help of the skin, which is called the oxygen diffuses out only through the skin. That is called the cutaneous respiration. Cutaneous respiration. Let us come to the next one, circulatory system. In arthropods, here I am comparing between the annelids and the arthropods. In annelida, you have closed circulatory system and you have this kind of tubes present, whereas in arthropods we have open circulatory system, and this is the space which is present. This is called the open circulatory system in cockroach. When we read more about the cockroach in Periplanet Americana, we will come to see about the circulatory system. We have a diagram here. Now look here, you can find that the body is divided into different groups. There is a different cavities because of this layer here. There is a septa present which divides here, and there is a space present here which is called the sinus. The vacant space is called sinus, and there are different cavities here which is filled with the hemolymph. Hemolymph. I'll magnify this and I'll show it to you. Look here, the body cavity, it is divided into three parts here. Where the heart is present, that is called pericardial, and then you have the visceral sinus, and then you have the some perineural sinus. All the sinus is made up of cavities which are called the sinus, and the fluid which is flowing is a hemocoel. There is a heart present. This is the heart present, which is 13 chambered in cockroach. So heart, you have a heart, you have fluid present, you have a cavity present, but you do not have blood vessels. When we talk about circulatory system in arthropods, always remember there are no blood vessels. That is the reason why it is called the open type of circulatory system. Now let us come to the reproduction in arthropoda. In arthropoda, reproduction, we can clearly identify a male and a female. A male cockroach and a female cockroach, they have an external morphology due to which we can easily identify them. So sexes are separate in arthropods, whereas when you read about an annelida, earthworm, it is an hermaphrodite. I am telling this so that you can understand the difference here. Here it is separate. So there are both male and female individuals are present. Then they have a paired sex organs are present which produce gametes. Here the gametes are produced. These gametes are the ova and sperm. Ova and sperms are produced. Sperms are in the form of a group. There are many number of sperms which are released in a bundle or a sac, that is called the spermatophore. I will magnify this and I will show you once again, but then you have to remember here. Next one, the arthropod eggs. They are very rich in yolk. So what does that mean? That means that when there is more amount of yolk, these eggs are laid and they are independent. So yolk should serve as a nutrition for the developing embryo or the developing larvae of embryo. So there is lot of in egg present. So most of the time they are oviparous. Oviparous. So they lay down the eggs. But sometimes what happens, if some animals, they show parental care. They show parental care, and in that parental care, this viviparity is shown. Viviparity is shown. So the animal will give birth to the young one, and this is seen very much prominently in wolf scorpion. Wolf scorpion, it tries to carry this organism on its back. Next one, now let us look at the egg after fertilization. One more exception that is seen only in arthropods. Arthropods, that is parthenogenesis. When you look at the how honeybees, when unfertilized eggs, if they are fertilized, they will form the zygote. Ova and sperm, then when they fertilize, they will form the eggs. They will form the zygote. But here the zygote is formed because of unfertilized eggs. That is called parthenogenesis. And that parthenogenesis, due to parthenogenesis, unfertilized eggs, what is happening is the organism that is formed is a worker or a unfertilized female is formed. We will come to that when we read about the organisms in special. Now let us come to the formation of many number of eggs, that is the unfertilized eggs. Now let us look at the larvae. Each stage, when I talk about the classification, I will tell you about I, I will tell you about the different kind of I will tell you about the different kinds of larvae. But here what we are going to read is, the first thing you have to remember is that Noophilus larva is the first larva of arthropods. There are many different larvae. Zoea larva is there. There different kinds of larvae are there. But then we are this larvae you have to remember because of the arthropods. Now after this, let us come to the next slide. Ecdysis. The development when there is sometimes the zygote, it gives rise to the adult, which is same. This kind of, they can either give rise to the adults which are similar in shape, or with the zygote which are different. You look at the life cycle of butterfly. In butterfly, the young one or the larvae is caterpillar, but the adult is different. Here I have taken the example of aphid. Whereas here let us look at the another one. This is the example which we are taking here. From the zygote, you will get the nymph. This is called as the nymph. I will run this slide once again. I'll revise it in the next session once again. But remember that the zygote, when it hatches, it forms the nymph, and it is similar to the adult. The classification which I am going to take later in the next session is about arthropoda, which is divided into Three phyla, Trilobita, Chelicerata, and Mandibulata. Again, we have the different classes which I am going to continue in the next session. With this, I am very much thankful to everyone who has given support to make this session successful. Thank you very much, madam. I'll end the
Thank you very much, Suneela Madam, for the extensive explanation of the Arthropoda phylum. I'm sure all students must have gained a clear understanding of Arthropoda characteristics. Tomorrow, we will be going through Arthropoda classification and complete the study of phylum Arthropoda. I request all the students to read the textbook chapter thoroughly, watch this video again, and make notes. Thank you, Madam, for this presentation, and thanks to all the students for attending. And I hope all students find these sessions useful and continue to participate in large numbers in the future as well. Students may stay subscribed for regular updates. And thank you very much, Madam, once again. Thank you.